Hey, good day, everyone. Welcome to the STEM Lockdown Digital School. This is grade 11 Life Sciences, and my name is Onkarabetse Setso. You guys can get hold of me on my Twitter handle, which is at Onkarabetse_ace. Okay. As you guys know, since the beginning of the week, we have been looking at the human digestive system, and we will be continuing from where we left off. But
Bali, you still can't hear me?
Sound is better, ma'am.
Is it okay now?
Yes.
All right. Okay, cool. All right, guys. As I said, that we will be continuing from where we left off yesterday. And before we do that, let us just quickly go through what we dealt with on Tuesday. Tuesday, we were looking at the difference between the alimentary canal, right, and the accessory organs. So we did say that the alimentary canal is from your mouth to your esophagus, your stomach, your small intestine, your large intestine until the anus. And we did say that in this picture here, it is depicted by this color pink, right? And then we also looked at our accessory organs. That was Tuesday, our liver, the gallbladder, the pancreas, and the salivary glands, right? And yesterday, we looked deeper into digestion. So the type of digestion that takes place in the mouth. We said that in the mouth, we have chemical digestion happening, and we also have mechanical digestion happening. We looked at the functions of the tongue. I hope you guys can recall that the functions of the tongue. We also looked at the salivary glands and what their function is in digestion. Remember, guys, we also mentioned an important enzyme that is released by the salivary glands, and we said that enzyme is amylase, right? And it helps in the breaking down of carbohydrates or starch. And then we dealt with the area here, which is called your pharynx. We also said that the food bolus, okay, it is pushed to your esophagus, or rather goes to your esophagus and not your breathing pipe, because your breathing pipe, when you swallow, the epiglottis closes the breathing pipe. I hope you guys can remember that. And we said that the food bolus moves down to your stomach via peristalsis. Okay, and we got to the stomach and we spoke about mechanical digestion happening in the stomach. We also spoke about the chemical digestion that takes place, the enzymes that are released, and that now once the food is mixed, we have a liquid, an acidic liquid called chyme. Okay, so we are going to continue from there, from how our chyme moves from our stomach to the small intestine. But also before that, let us just quickly revise a few definitions. Ingestion, we said that this occurs when food is taken in by the mouth. The second thing was mechanical digestion is the crushing and shearing by means of teeth. I hope you guys can remember the 4 types of teeth that we have as humans, which are your incisors, your canines, your premolars, and your molars. And I hope you guys remember the function of each. Okay. The third thing is chemical digestion, the chemical breakdown of food. Then we have absorption, which is the movement of digested products, right, into the blood. Those are your vitamins, minerals, and water, and that is what we'll be mainly looking at. So absorption is the movement of products of digestion into the blood, okay? So you guys just need to keep this definition in mind, because remember Tuesday when we dealt with the whole alimentary canal, we said that absorption takes place at the small intestine. Yeah. And the fifth thing is assimilation. This is the uptake and the use of glucose and amino acids by the body cells for energy and various functions. And the last thing here is egestion, which is the removal of undigested food in the form of feces. Okay, so let us just quickly look at our study objectives. First one, you should be able to explain what is meant by digestion and also state which parts of the small intestine are involved with digestion. Okay, so we need to explain digestion and also state which parts of the small intestine is involved. We need to be able to state the substrate and products which are involved with the following enzymes: carbohydrates, proteases, and lipases. Then we should also be able to define absorption. We'll just give a simple definition of absorption. We just dealt with it now, and explain how it takes place in the small intestine. Then we need to explain there, right? And then we also need to be able to draw the structure of the villus. Remember, when we draw structures, we also need to be able to label the structures. Okay. So guys, I will need you guys to use the Zoom group chat for any questions or answers that you guys have. And if I am going too slow, or I mean too slow or too fast, just please indicate for me on the group chat. And just also another thing to note that throughout the lesson, there will be a whole lot of repetition because guys, some of these concepts are quite hard to grasp. So yeah, just bear with me. Okay, the small intestine, right? So the small intestine is divided into 3 parts. As you guys remember from yesterday's video that we watched before the lesson, that they indicated on the video that we have the duodenum, the jejunum, and the ileum, all right? So those are the 3 parts of the small intestine. It is approximately 4.5 meters long in a living person and 6 meters long in an autopsy, all right? So that means the small intestine of a dead person is about 6 meters long, and that is because the muscles are relaxed. Okay. The small intestine releases carbohydrates. And guys, okay, another thing, please use the Zoom group chat. What are carbohydrates? I just need an answer from you guys so that I know that you guys are following. What are carbohydrates? And I'll just continue. Okay, the small intestine releases carbohydrates, right? And the first part of the small intestine, remember we said that it is the duodenum, that receives acidic chyme from the stomach when the pyloric sphincter opens. Okay, and another thing to note about the small intestine is that absorption and digestion takes place. What's important is both absorption and digestion takes place. Guys, I asked what is carbohydrates. Please use the group chat to send through your answers. Right, now let's look at the first part. So the first part of the intestine, of the small intestine, as mentioned, it is the duodenum. Okay, so guys, yesterday we dealt with the chyme that would be mixed here in the stomach. So now this needs to move into your duodenum. If you guys can just follow with me on the picture. So now in order for this chyme to move into your duodenum, the pyloric sphincter, here's the pyloric sphincter, needs to open up. So the moment it opens up, your chyme enters your duodenum, right? So it'll just move here. And that is just part of your duodenum. So this will be your chyme right there. Okay. What we also have in the duodenum is what we call the hepatopancreatic duct. And that is shown by this here. This is your hepatopancreatic duct, which opens up into your duodenum. So what is the hepatopancreatic duct? Basically, this is where you have your pancreatic duct. So you guys can just follow with me here. Your pancreatic duct and your common bile duct. So your pancreatic duct comes from the pancreas and your common bile duct comes from your bladder. So, and this is the part where they join at the hepatopancreatic duct, which opens up into the duodenum, right? As I said, that the common bile duct comes from the gallbladder. The pancreatic duct comes from the pancreas, right? Okay, Talia, enzymes that break down carbohydrates into monosaccharides. That is correct, and thank you very much for the answer. Okay, so we all understand what's happening here, right? We all understand what the hepatopancreatic duct is. And we also understand that it comes from the gallbladder and the pancreas. Okay. Moving right along. So digestive enzymes and bicarbonates are received from the pancreas. So that pancreatic duct, it brings in digestive enzymes and bicarbonates, all right? And then bile, which is your bile pigments and your bile salts, are received from the liver and the gallbladder. And the reason why we say the liver and the gallbladder, that is because, remember, we said that bile is manufactured or created and secreted in the liver, okay? And after that, it moves to the gallbladder, and that is where it is stored. So in order for it to be released into the duodenum, it comes from the gallbladder because it is secreted in the liver, then it is stored in the gallbladder, then the gallbladder releases it into the duodenum. I hope everyone is following. Okay, so the duodenum is involved in neutralizing the acidic chyme. Remember, the chyme that comes from the stomach has HCl. So HCl is an acid, and that is why our chyme is acidic. And it is also involved in the digestion of carbohydrates. Remember, we said that the small intestine also releases carbohydrates, okay? So in the duodenum, we also have the digestion of proteins using proteases, and we have the digestion of lipids using lipases. Remember, our carbohydrates are the enzymes involved in breaking down starch or carbohydrates. Our proteases are enzymes involved in breaking down proteins into its simpler forms, polypeptides, and then amino acids. Yeah? The products of, of proteins are amino acids. And then lipids are broken down using lipases. So after lipases act on substrates, we get fatty acids and glycerol, right? So those are the components of fatty material that we consume. Okay, so guys, yesterday we mentioned one enzyme which falls under a protease that is released in the stomach. What is the name of that enzyme? Guys can just answer in the group chat as we continue. So guys, in the duodenum, again, we said that digestive juices from the pancreas and the liver combine with chyme, making it thin and watery. Okay, then we move on, okay. So let me just wait for Divine so that the sound is fixed. Okay, please repeat the question. Okay, I said yesterday we dealt with an enzyme that is released in the stomach. It's a protease. So what is the name of that enzyme that is released in? Okay, all right, guys, are we following though? Are we following? I hear a student is saying I'm going too fast. Are we following? Or should I just repeat the previous slide? Should I repeat the previous slide or are we following, guys? All right, cool. Let me start here, okay? Because this is where everything starts. And to all of you guys who said pepsin, yes, that is correct. All right.
So we said that the duodenum is the first part of the small intestine, right? So when the acidic chyme is exiting from the stomach, it enters into the duodenum. Okay. And it can be indicated by what we have here. Let me just use a laser pointer. The acidic chyme enters there, right? That is where our pyloric sphincter is located, and that is the entry and exit point of the acidic chyme from the stomach to the small intestine, respectively. Okay. And then we said that here we have what we call the hepatopancreatic duct. Okay. So what happens at the hepatopancreatic duct is that the pancreatic duct is connected, and we also have the common bile duct, which is connected there. Yeah? The common bile duct, it comes from the gallbladder. Remember, we said that the gallbladder stores bile, and that bile is secreted by the liver. All right. So at this opening here, bile is secreted into the duodenum, and we also have pancreatic juices, which are also secreted into the duodenum through the hepatopancreatic duct. Okay, I hope everyone gets that point and what basically the slide was explaining. Okay, and then the other point that I mentioned was that digestive enzymes and bicarbonates are received from the pancreas. Remember in the previous slide, I said that pancreatic juices are released into the duodenum via the hepatopancreatic duct. So what that pancreatic juice is your digestive enzymes and your bicarbonates, which come from your pancreas and are entering your small intestine. Okay, so this is where we have a connection of your accessory organs and your organs of the alimentary canal. All right. Let me just pause until Michaela's problem is fixed. Okay, guys, am I clear? Can you guys hear me properly? Michaela, can you hear me properly? All right, cool. So the digestive enzymes and the bicarbonates form part of the pancreatic juices which enter your small intestine, right? Your duodenum, right? And then we said bile. Remember, we had a bile duct which comes from your gallbladder. Bile is received from your liver and the gallbladder. And I said that the reason why we mentioned liver here is because bile is secreted in the liver, but it is stored in the gallbladder. Therefore, when secretions happen, they happen from the gallbladder to the small intestine. Okay. And then I said that the duodenum, what happens in the duodenum is that we have the neutralizing of the acidic chyme, because remember, it will be mixing with the pancreatic juices and it will be mixing with the bile, right? So there will be neutralization that is happening by the small intestine at the duodenum, the first part, okay? And then we will also have digestion of carbohydrates using carbohydrases. Because of this point here, we have digestive enzymes and bicarbonates that are released from the pancreatic duct into your duodenum, okay? And then we'll also have digestion of proteins, and we'll have a digestion of lipids that are happening, that is happening in the duodenum. Okay, Musala, you can ask your question in the group chat. I see you raised your hand. You can just ask your question in the group chat. And then this last bullet here is basically just summarizing what is happening at the duodenum at this particular time. The digestive juices from the pancreas and the bile from the liver combine with the chyme, making it thin and liquid.
Teacher, please check your sound.
Okay. Can you guys hear me? Is the sound okay now? Can I just get an indication, please?
Sound is okay?
Thank you. All right, so that just made up the duodenum. This is where we end with the duodenum, which is the first part of the small intestine. Now we move along to the duodenum, which is the second part of the intestine, right? So by the duodenum, it is mainly just involved in digestion. There is no ducts which bring in other enzymes from other accessory organs or anything like that. So the digestion that takes place by the duodenum is chemical digestion, because we have our carbohydrates, the proteases, and the lipases that were secreted or released into the duodenum. All right, are we all following? Okay, so and guys, remember, we also have the third part of the small intestine, which is called the ileum, right? And by the ileum, it's mainly absorption that takes place there. Okay, so I just didn't cover it here because it's just absorption that takes place there. Okay, cool. So as we continue, let's look at the structure of the alimentary canal so that we understand how this absorption takes place. So we have 4 layers, right? We have 4 layers. Let me just, okay, I can just use this. We have 4 layers to our alimentary canal. The outer layer, as you guys can see here, it's your brownish color, is called your serosa, all right? Which is later followed by the muscle layers, which have your longitudinal muscles and your circular muscles. So this is where contraction and relaxation happens by your muscle layers. And then we have your submucosa. We have a submucosa here. So the submucosa, it has, okay, wait, before we get to the submucosa, let me just deal with the muscle layers here. By the muscle layers, we have your longitudinal muscles and your circular muscles, and we also have what we call connective tissue, okay? So the connective tissue is your blood vessels, it is your lymph vessels, and it is also your nerves, all right? Then we can move on to your submucosa, which is followed by the mucosa here, right? So the mucosa, that is your innermost layer. So this is the place which will receive your chyme, right? And it has goblet cells which release mucus. So this mucosa here is a very moist layer because there is secretions of mucus happening at the mucosa. Okay, so that is, I think, yeah, this covers what I have explained. We did say that, okay. We dealt with that. And then we also, okay, I didn't deal with this. So the mucous membrane has millions of tiny finger-like projections. And these finger-like projections are called villi. So the function of a villi is to increase the surface area for the absorption of food. And here is a picture of the villi. Here is a picture of the villi, guys. So your small intestine is like this, and this is where your serosa is. That is your muscle layer, your submucosa, and your mucosa. All right, so this is how your small intestine looks like in the inside. And let me just get a pen. And this is where your chyme will be, all right? And all the other digestive enzymes which are released and all those juices. So this is where they will be at, right? So you guys remember that you guys also need to be able to draw the structure of the villi, but don't draw it now. I'll show you guys a simpler structure of the villi as we move through with the lesson. Okay, so what happens at the villi? This is where absorption of nutrients takes place. So basically absorption of the digested food. What we have at the villi, here is a close-up. We have epithelial cells, which are usually referred to as columnar epithelial cells. We have what you call your lacteal, which is there. And we have your capillaries, right there. So this is blood going in, and this is blood flowing out. Okay. And your lacteal, it is connected to your lymphatic vessels, right? And what we also find in the villi is that it has microvilli, all right? That is just for an extension of the surface area to allow for more absorption to take place. So this is a structure of the villi. I hope we're all following, guys. If you're not following, just stop me. If there's anything I need to repeat, just mention it, okay? Okay, so now absorption of food at the villi. So the villi, they are found throughout the small intestine. So basically from your duodenum until the ileum. Okay, and absorption, which is important, absorption of nutrients happens there. So we have the absorption of nutrients happening there, either by diffusion or active transport. And guys, remember, in order for active transport to happen, we need ATP. Therefore, there has to be mitochondria somewhere. Okay, so these epithelial cells here, they're also filled with mitochondria, right? I hope you guys remember the function of mitochondria from grade 10. They have mitochondria around them. That is for active transport to take place. So the nutrients, they will travel via the epithelium, right? They will, from your chyme, they will travel via your epithelium to your blood capillaries, right? So what are these nutrients that are being diffused or traveling via active transport from your epithelium to your blood capillaries? Those are our amino acids, simple sugars like glucose, fructose, and we also have some fats. Some fats also actually go through the epithelium into the blood capillaries. And then our fatty acids and glycerol, they enter the lacteal, right? Which is connected to your lymphatic system. And then they finally go into the bloodstream. Okay. Please repeat the process of the villi. Is it from this slide or from the previous slide? Okay, let me just start here. Okay. Remember I said that this is our small intestine, right? That is the tube, the alimentary canal tube. Okay. So here we have our serosa, we have our muscle layer, we have our submucosa, and we have our mucus layer. So by the mucus layer, remember here, this is where we'll have the chyme coming in from the stomach and in the intestine, and we'll have released digestive juices and enzymes here, right? So they are in contact with your mucosa. And what is found on the mucosa is that we find a villi, right? Your villi, they are found on the mucosa. Here are your villi, right? And they are in contact with the digestive juices in your chyme, right? So from that, we need the nutrients, the products of digestion to get into our bloodstream. So that is where the villi plays its role, because it has the epithelial cells, which is just one layer, right, of cells, and it allows and assists the nutrients to actually get into the blood capillaries, right? So the nutrients will move from your chyme, right, through your epithelial layer here into your blood stream. So to your blood capillaries, or it will go to your lacteal, which is present here, and it is also here. Marcella, do you understand now? So what is being said here is basically what I have just explained on the previous slide. All right, cool. Okay, so remember we mentioned the epithelial cells, the columnar epithelial cells, which could be indicate, let me just get a color that we'll all see. And they are indicated here. So these are your columnar epithelial cells in this diagram here, and they function to absorb monosaccharides, amino acids, fatty acids, and glycerol. So those are our nutrients that we need in our blood. Okay. And then we have the capillaries, which carry away water-soluble substances. This means that it is the nutrients that can dissolve in water. So these are nutrients like your vitamin B, your vitamin C, your salts, and certain monosaccharides, okay? They are taken into your bloodstream, right? And then the lacteal, which is this one here, it carries away fat-soluble substances. So your vitamin A, D, E, and K, they are absorbed into your lacteal, right? Because they are fat-soluble and can dissolve in fats, right? So guys, here is a simple structure of the villi that you guys need to be able to draw. So you guys need to indicate the epithelial cells, which is a thin layer and allows for faster absorption of the nutrients. You guys also need to indicate capillaries, right? So blood coming in and the blood going out. So remember here we'll have the absorption happening from the epithelium. So monosaccharides will enter, your water will enter, your amino acids will enter, right? And which means that the blood going out will have more nutrients than the blood coming in, okay? And then we also have another structure there, which is the lacteal. Okay, the lacteal, which takes in your fat-soluble substances, right, guys? So just use this picture here in order for you guys to be able to draw the villi. This is a simple structure of the villi. And then another important thing here that we can see is that the blood goes to the hepatic portal vein. All right, so this is the blood after absorption, the blood that is full of nutrients, the blood that has our amino acids, that has our monosaccharides and some fatty acids. It goes to the liver. All right, and we will deal with the hepatic portal vein next week. So do we all understand what happens at the villi or the villus? Do we all understand what happens at the villi? All right, cool. Okay, so here is just another structure. If you guys didn't get what happens at the villi from the previous slides, you will probably get it now. So we have a thin epithelium, right? Here are your epithelial cells that we spoke about. And we also said that they are rich in mitochondria. This is where diffusion and active transport takes place. Diffusion and active transport of the nutrients, right, of the digested food. So the skin of each villus is only 1 cell thick, and this allows absorption to happen very fast. And then lacteal, which is the structure here, absorbs fatty acids and glycerol. And then we have a good blood supply. The blood capillaries absorb simple sugars and amino acids. Okay. And remember that this lacteal goes to your lymphatic system, and that blue vein there goes to your hepatic vein portal system, which I said we will deal with it next week. Okay, the pancreas. Okay, guys, let's go back to the duodenum. What happens at the duodenum? There by the hepatopancreatic duct. Remember we said that at the hepatopancreatic duct, digestive juices or pancreatic juices are released from the pancreas, right? Meaning that the pancreas can be seen as an exocrine gland because it utilizes ducts, okay? And it secretes pancreatic juices with enzymes. Mbonge, you can ask your question in the group chat. It secretes pancreatic juices with enzymes. These enzymes include trypsin, it includes pancreatic amylase, and it includes lipases. Okay, so trypsin gives us amino acids, meaning that it breaks down proteins. Pancreatic amylase gives us monosaccharides, which means that it breaks down carbohydrates or starch. And lipases digests fat. Okay, so that will give us fatty acids and glycerol, right? And another thing about the pancreas is that it can also act. Okay, what is the pancreas? Okay, the pancreas is an accessory organ, which is, I think I have a picture here. It is an accessory organ which is involved in digestion. So this is the pancreas. Here is the pancreas muscle. All right. Are you sorted? That is the pancreas. It's an accessory organ which is involved in digestion. All right. Cool. And another thing about the pancreas is that it can also act as an endocrine gland. This means that the pancreatic islets are utilized and it secretes hormones, namely insulin and glucagon into the bloodstream, and it regulates blood sugar levels. So we will look at the functions of the pancreas as an endocrine gland when we deal with diabetes. Okay. Okay, there's a question which says, what is trypsin? Trypsin is an enzyme which is released by the pancreas and it digests proteins. So it breaks down your proteins into polypeptides and into amino acids. That is what trypsin is. Okay, and here is the pancreas. And this cross section here is showing us the pancreatic islets of Langerhans. These are the ones that are involved in the release of insulin and glucagon, and we will look at this later, but this is the pancreas, guys. Okay, now let's look at the gallbladder and bile. So bile is secreted in the liver, but it is stored in the gallbladder. Remember we mentioned that, ne? And then bile salts prepare fats for subsequent digestion. So they work like detergents and they disperse large droplets of fats. For example, if this is a fat molecule, when bile salts act upon them, then it will give us something like this. Just smaller pieces of fat, right? It will just break down the bigger circle into smaller pieces. Cool. So bile pigments are produced in the liver from dead red blood cells. Bile pigments, guys, they also form part of bile. So bile has bile salts and bile pigments. Bile pigments is waste products from the liver, which are being excreted. Ne? These ones don't play a role in digestion. They don't play another role in digestion. They also want to go to be part of the feces that will be excreted. Okay. And then the gallbladder stores bile. When bile is too concentrated, gallstones may form, and these will be flushed through the bile duct and then excreted. Right. So your gallstones are just solids which are formed by mineral salts and water. Okay. Okay, so in this picture here, this is actually bile. I'm sure that you guys can't see it. This is bile. And this is an arrow showing us that the bile is going to the gallbladder, right? And then we have another arrow here, which shows the bile going through the common bile duct and is going to be secreted here into your small intestine. Okay. So basically that is the connection between your liver and your gallbladder. And the other functions, we already dealt with them. We already dealt with acid chyme entering your small intestine, and we also spoke about the pancreatic juices entering your small intestine, right? Okay, the liver. So what happens at the liver? It secretes bile, as we have seen from the previous image. It receives all end products of digestion via the hepatic portal vein. And guys, this bullet, let it not confuse you. We are going to deal with it on Monday, right? Excess glucose is stored in the liver. Excess amino acids are deaminated in the liver, and alcohol and drugs are detoxified in the liver. Okay, so we're gonna look at the liver in greater detail on Monday. There's now questions, guys. The duodenum, ileum, and jejunum form part of A, small intestines, B, large intestine, C, stomach, D, esophagus. Can I get answers, please? All right, so we're following. It is A, small intestine. Next question. The outermost layer of the wall of the small intestine is A, serosa, B, muscular layer, C, submucosa, or D, mucosa. All right, beautiful. It is the serosa. Remember, the mucosa is the innermost layer, right? And that is where we find our villi, right? And your submucosa is the next layer. That is where we find our connective tissue. Your muscular layer has muscles, and your serosa is the outermost layer. Okay, and here's another question. Let me just quickly check time. Okay, guys, time is almost up. So I'm gonna give it to you guys for homework. Okay, so just take a screenshot of this question and complete it. Tomorrow, not tomorrow, sorry. Monday, we will go through the answers of this question. So it reads, the diagram below illustrates the microscopic structure of a villus. Study the diagram and answer the following question. So here we have the structure of our villus. Remember that this is found where? On the mucosa, all right? That is on the innermost layer of your small intestine. Question 2.3.1, it says identify the part labeled 3 and its function. Question 2.3.2 says which labeled part contains blood with relatively higher amounts of glucose and amino acids. Question 2.3.3, name the process that enables humans to absorb nutrients mentioned in question 2.3.2. And question 2.3.4, explain how the villus is structurally adapted to enhance the absorption of digested nutrients from the small intestine. So guys, I hope you all have written the question or have taken a screenshot. Tomorrow, we're going to go through the answers, and not tomorrow, sorry, Monday, we're going to go through the answers, and you guys are going to give me the answers. Okay, guys, thank you very much for joining us today. And I hope you guys understood absorption taking place in the small intestine. Remember to go through the work again throughout the long weekend, study, and I will see you guys on Monday. Bye-bye.